Welcome to Chapter 5 on metabolism. Metabolism is the sum of all chemical reactions that occur inside of an organism. And we can break that down into two types. So there's catabolic pathways or reactions that are going to be breaking down large macromolecules into smaller, more simpler ones. And through that process, it's going to release energy. On the other hand, there's anabolic pathways, which are going to now take those smaller molecules with the use of energy and build them up into larger macromolecules. Catabolic pathways would be like breaking glucose down into carbon dioxide and energy, whereas anabolic pathways, an example would be taking amino acids and putting them together to make a protein, and that process is going to require energy. Now, both catabolic and anabolic pathways are then linked. So the energy is released through catabolic pathways, and then that energy that was released is then used in anabolic pathways to build up other molecules. So here's another example of that. So if we start with complex molecules, like starch, and then we go through catabolic reactions, that's going to release or produce energy in the form of ATP through the catabolic reaction that's forming smaller molecules. And then we can take these smaller molecules and build them up through anabolic reaction using ATP to build complex molecules. So that's the coupling of catabolic and anabolic reactions. So chemical reactions, as you recall from when we talked about in Chapter 2, involves the process of atoms filling up those valence shells. They fill up those valence shells with more electrons. That makes them more stable. A couple of terms. So in order for a reaction to occur, there's a certain amount of energy that is required in order to cause that reaction to occur, and we call that activation energy. A reaction rate is the rate in which these reactions are occurring, or the rate in which products are formed. A reaction rate can vary. It can be increased through the use of enzymes, which we see commonly in the body. Reaction rate can also be increased through increasing temperature, which we don't see too much in the body. It's pretty consistent, right? And also through increasing substrate concentrations. So here we're looking at activation energy. What we're looking at is the level of energy throughout time as we move across throughout the reaction. So here's the energy level of the reactant. There's a certain amount of energy, which we call activation energy, that's required for that reaction to occur. Now, what enzymes do, enzymes help to lower the amount of energy that's required. So enzymes help to lower the activation energy. And it does this by bringing the substrates together or by helping to break them apart. So why are we talking about this? Why is metabolism important at all when we're talking about microbiology? Well, first of all, there's a lot of benefits that we have from microbes. I think that when we get so focused on in the medical field, we tend to think of microbes as the bad guys, but there is a lot of benefits to them, and their metabolism is a huge part of that. So through microbe metabolism, we can produce vaccines. Also, the microbes in our gut, they help to break down nutrients, and they help to release certain vitamins that we would not be able to get on our own. And it's through their metabolism that they're able to do that. Also, different enzymes that we use is through microbial metabolism. Other reasons to study this would be my microbial metabolism is an excellent target to use for different drugs. So now we're looking more in the medical field, and when we're trying to kill or reduce the growth of bacteria, we can target their metabolism to help kill them or reduce their growth. Also, different microbes have different metabolic pathways. And by identifying the metabolic pathways that's carried out through those bacteria or different microbes, that can help us in identification. And when we identify a species, that can help with patient treatment. So we're going to see that come back when we start looking at biochemical tests in the lab. That's what we're looking at, is we're looking at the different metabolic pathways of the different species when we're trying to identify them. So keep all that in mind as we move forward. Metabolism might get a little bit high detailed, might get a little bit confusing, but keep that in mind. That's why we need to understand metabolism. Also, some key concepts about metabolism. It's enzymes actually that are facilitating these metabolic reactions. So that's where we're going to start. We're actually going to start by talking about enzymes. Also, ATP is another big one. So ATP is used by all cells for energy use. And so then so our next step is we're going to look at ATP. Then as we already discussed, we have catabolic reactions and anabolic reactions. So getting started with our enzymes. So enzymes, as you know, are biological catalysts. They are very specific. They're very, very specific. So each enzyme is only going to carry out one reaction, and it's very specific to the substrates that that enzyme can bind to. And it's like a lock and key. So that substrate shape is specific to fit into the enzyme site here. Also, enzymes are unaffected by their reactions, so they can be reused over and over. So a couple of different sites that you guys need to know. So this green thing here, that's the enzyme. The red part is the substrate. The substrate is going to fit specifically into what we call the active site. So that's where the substrate is binding to, is in the active site. There's another site called the allosteric site. This is where other molecules, which can include activators or inhibitors, are going to bind to. So substrate binds to active site. Other molecules are going to bind to the allosteric site. The names of enzymes, so they're pretty much always going to end in ase. So if you see a term that ends with ase, that's an enzyme. So I want you guys to know that moving forward. Know that anything that ends with ase is an enzyme. The rest of the name is often going to identify the substrate. That's not quite as hard fast of a rule as the ase part, but it's pretty typical. So like DNA ligase, a couple of examples. DNA ligase is going to like stitch together DNA. Sucrase is an enzyme for sucrose, etc. So some things that influence enzyme activity. Enzymes are proteins. Remember when we talked about proteins, we talked about how proteins can be denatured by changes in temperature and pH. So that happens when temperature or pH changes. That's going to affect the bonds. See, here's an alpha helix. Here's another beta sheet that we talked about earlier in chapter 2. The bonds that make this shape will change when we change temperature and pH. And when that changes, that's going to change the shape. If the shape of the enzyme changes, it no longer can function properly. So here are a few different graphs of how enzyme activity changes as the temperature, pH, or substrate concentration changes. And I do want you to understand these three. So we're going to go through them. So here we're looking at increasing temperature as we move left to right. As you know, rate of reaction is going to increase as we increase temperature. And so that's going to hold true here as well for enzyme activity. If we increase temperature, that's going to increase the rate of reaction with the enzyme until a point. And there becomes a point in which that enzyme is going to now denature. It got too hot and it's going to denature. And then that is when enzyme activity will very rapidly drop off. So for temperature, we have a slow increase as temperature increases, and then the enzyme will denature because it got too hot, and then that it will no longer function. Each enzyme for pH has a specific pH that it is the most effective at. And as you move further away from that pH, it's going to become less and less effective. So this particular example, this enzyme is most effective at pH of 7, which is a neutral pH, right? So as we go more acidic or more basic, it's going to become less and less effective. The third one is substrate concentration. As we increase substrate concentration, it's going to increase the rate of reaction. That makes sense, right? We have more substrate. That's going to allow the enzyme to bind to more substrate and carry out more reactions until we reach a point. And we call that point the point of saturation. At the point of saturation, all of the enzymes are functioning as quickly as they can, and they can't go any faster. So think of it like you're working at McDonald's and you just have a couple of customers come in, and then you can serve them pretty quickly. You get more and more customers, you pick up the pace, you can work faster, until at some point, everyone that's working is working as fast as they can, and they just can't serve any more customers any faster. That would be the point, what we are calling the point of saturation. Enzymes can't work any faster, so that's as fast as the reaction rate is going to get. All right, so we also have inhibitors. So these are going to inhibit. There's different molecules or substances that will inhibit the function of an enzyme. There's two different types. There's competitive inhibitors and allosteric inhibitors. Competitive inhibitors are going to compete for, so here I think we have it labeled, yeah, competitive inhibitors are going to compete for the active site. So here's our enzyme in green, substrate in red. Here's a competitive inhibitor in yellow. So the substrate would like to come in and bind to the active site here in the enzyme, but it can't because the competitive inhibitor has instead bound and blocked that site. Whereas the allosteric inhibitor instead is going to bind to the allosteric site. What happens when something binds to the allosteric site is it changes the shape of the enzyme. So if you have something bound to the allosteric site, the enzyme has now changed shape so that it's not a perfect fit anymore for the substrate. So even though there's nothing bound here, the substrate can't bind because it doesn't fit anymore because the enzyme has changed shape because of being bound to the allosteric inhibitor. An example of a competitive inhibitor is sulfanilamide. And this is an antibiotic that serves as a competitive inhibitor for the enzyme that makes a folic acid in bacteria. So it's going to compete for that active site in that enzyme. It's going to block those enzymes. So now the enzymes in bacteria can no longer make folic acid. Without that, they cannot reproduce. An example of allosteric inhibitor is feedback inhibition. Now, this can actually be beneficial. Oops, sorry. And this is a part of what an organism wants to do. So often we see these big long pathways in order to make a end product. So molecule 1 undergoes this reaction to become molecule 2, undergoes this reaction to become molecule 3, and then finally it becomes its final end product. Now, eventually, we might have enough end product produced. So we don't need to make any more. It would be a waste of time, of effort, a waste of resources to continue to make this end product for the organism. And so what will happen is the end product will serve as an allosteric inhibitor on the first enzyme of the pathway. So that's going to signal to this first enzyme, don't continue on in the production of this product. We don't need any more, we have enough made. We see this not only in bacteria, but in all organisms, us as well. So this is very beneficial to help to limit the production of these end products. So if we have a lot of the end product, it's going to bind to the allosteric site and inhibit the production of more end product. Now, if the concentration of the end product begins to fall, then it will release from the allosteric site, and then that enzyme will become activated again, and it will start to make more end product. It's a good system, and it's called feedback inhibition. All right, switching away from enzymes. Now we're going to talk about redox reactions. First of all, oxidation is the loss of electrons, and reduction is the gain of electrons. It's 100% okay if you have to think. Leo says grr. I still have to think of it too, so it's okay. Leo is loss of electrons, or loss of electrons is oxidation. Okay, loss of electrons is oxidation. It's Leo. And then gain of electrons is reduction. Grr. All right. Now we can pair those two together, which we see quite often. So one molecule is going to be oxidized and the other will be reduced. So here we have molecule A is going to lose an electron. So it's oxidized. And molecule B is going to gain an electron, and so it's reduced. That electron is being transferred from molecule A to molecule B. That's what a redox reaction is a pairing of oxidation reaction with a reduction reaction, like we just saw here. We see this quite often in biological systems. So in this example here, this organic molecule has 2 hydrogen atoms. Remember, a hydrogen atom is simply a 1 proton and 1 electron, right? And then here we have NAD+. Through this reaction, 2 of these hydrogen atoms are going to be released from the organic molecule and transferred. We're going to get 1 complete hydrogen atom transferred, and then with just 1 electron. So since we're actually gaining a net charge of 1 electron, that's why we say that it's reduced. And then we just have the hydrogen proton is on its own. So those are oxidation reduction reactions. Here's another example of a redox reaction. This is aerobic cellular respiration, which is what we're about to start talking about. And what's happening here is we're breaking down sugar. So a glucose is oxidized to become carbon dioxide, and the oxygen is instead reduced to become water. So the oxygen is going to be gaining electrons to be reduced to water. All right. Quickly, before we get more into that, we're going to look really quick at ATP. So phosphorylation is the adding of a phosphate to to anything, but in what we're talking about right now is we're adding that phosphate to ADP. When we take off a phosphate, remember ATP has 3 phosphates. When we take off that last phosphate, that's going to release energy. So when we break this bond here, it's releasing energy. Okay, there's 3 different ways that phosphorylation can occur. This is how we're making ATP right now, 3 types. Substrate level phosphorylation. This is when we are simply going to take a phosphate and add it to ATP, or ADP, sorry. So we take ADP, add a phosphate, that's coming from a substrate, so any kind of substrate. We're going to take that phosphate off that substrate and add it to ADP, and that will give us ATP. That's called substrate level phosphorylation. Second type is oxidative phosphorylation. We're going to talk about that here in a little bit. Well, oxidative phosphorylation is carried out in the electron transport chain. And the third type is photophosphorylation. We're not going to talk about that today. I just want you to be aware of it. And it's carried out by converting light energy to ATP. All right. So there's 2 different types of energy production. The first one is cellular respiration. This one's carried out in aerobic environments, so it's going to be using oxygen. The second type is fermentation, and that is carried out in anaerobic environments, so environments where we do not have oxygen. First, we're going to talk about both of these. First, talking about cellular respiration or aerobic respiration. I do want you guys to watch these videos on your own. I'm not going to watch them here. We're going to move on to aerobic respiration. There's 3 steps. Glycolysis, which is the oxidation of glucose to pyruvate. Krebs cycle, oxidation of pyruvate to carbon dioxide. And the electron transport chain, which is the 3rd step, which is the oxidative phosphorylation, which we just saw was one of the ways to produce ATP. All right, so first glycolysis. So glycolysis is taking a glucose, it's going to break it down into pyruvic acid. At first, it's going to actually take 2 ATP to get this process started. Then it's going to release 4 ATP and 2 NADHs. That's mostly what I want you to get out of all these steps. It's just, what are we starting with? What do we get in the end? And then what forms of energy are produced? This NADH here, was starting with NAD+. Remember when we were talking about oxidation and reduction and about how it's taking those electrons. So the NADH is being reduced and it's taking those electrons. So think of NADH as this little guy carrying electrons around, okay? So he's just going to carry the electrons. He's going to carry them to the electron transport chain, which we're going to talk in a little bit. So just put that on the side of your mind there when you see that NADH. So that's glycolysis. Next step is Krebs cycle, but before the Krebs cycle, there's this transition step. It's taking pyruvic acid and making acetyl-CoA. Basically, it's removing a carbon dioxide and making an NADH in the process and adding this CoA. Then in the Krebs cycle, we're going to break everything down all the way to carbon dioxide. We're going to get 3 NADHs out of it, an ATP, and an FADH. And this is for each pyruvic acid. Remember, there was 2 pyruvic acids made for each glucose. So if we look at the whole thing for each glucose, it's going to be 6 carbon dioxide, and this is including the transitional step. We have 6 carbon dioxide, 8 NADHs, and 2 FADH2. FADH2, basically the same thing as NADH. It's just a little bit different molecule, same function though, it's carrying electrons. All right, so far what we've done is we've taken glucose, we've broken it down, we've taken all the electrons out of it, and we've transferred it to these NADHs, also that FADH2, and they're carrying the electrons. They're going to carry those electrons here to the electron transport chain. And they're going to dump those electrons into these proteins. They're going to move from protein to protein. Eventually, those electrons are going to reach oxygen. And so then once the electrons are added to oxygen, it's going to make water. Now, every time these proteins are getting an electron, it's going to pump a hydrogen ion across the membrane here. So this will either be in a eukaryotic cell, this will be carried out in the mitochondrial membrane. In a bacteria or a prokaryote, it's going to be carried out in the cell membrane, right? Because they don't have mitochondria. So now we're producing this hydrogen gradient. Now that we've made this hydrogen gradient, hydrogen has a desire to come inside the cell. It's going to do that through this ATP synthase. And it's the movement of hydrogen ions through the ATP synthase, which is going to power and produce massive amounts of ATP. So that was what all this was all about. We're taking those electrons so that the electrons can power the gradient, so that the gradient can power the ATP synthase, which makes ATP. All right. Here's just a summary of everything that we talked about, what's produced in each step. I do think you guys should study this. It talks about each step and what's produced. Again, we have step 1, glycolysis, breaking glucose down to 2 pyruvic acids. Then those are going to enter through the transitional step into the Krebs cycle, finally breaking everything down into carbon dioxide. Now we've produced all of these electron carriers, which will then go to the electron transport chain, which through the production of the gradient will then power the ATP synthase, making massive amounts of ATP, which was our final goal. Go through these 2 slides on your own. If you have any questions, let me know. Just helping you review where all this is happening, what's produced, when, and where. I think you guys will get more out of it if you do it yourself. So this whole process, we mostly think of sugars and carbohydrates as powering this whole process of making ATP. And that's typically what's used. But if necessary, other substances like proteins and lipids can be used. So what happens there is it's going to break down those proteins and lipids into smaller substances, which can then be added into this process at various points. All right, so that was cellular respiration or aerobic respiration. Now we're going to talk really quick about fermentation, which is anaerobic respiration. Respiration carried out in an anaerobic environment. So remember, we had that oxygen. That was the final electron acceptor in the electron transport chain. If we don't have oxygen to accept those electrons, then it doesn't function. The whole process is pointless, right? There's no point in making all of these NADHs to carry electrons to the electron transport chain if those electrons have nowhere to go. So instead, we have this other process, which is still going to use glycolysis. So that step is exactly the same. So we still have glycolysis, which is going to produce 2 ATP. As you notice, it's a lot less ATP. It's a lot less effective than cellular respiration. But if you don't have oxygen, it's something that these bacteria can carry out. So again, we're taking glucose, making pyruvate through the process of glycolysis, making 2 ATP and 2 NADH. So nothing's going to change there. The only thing that we're going to add now is we're going to add another step that's going to produce a final end product. And most commonly, we see lactate or ethanol. There's other end products that can be made as well. But the purpose of making this end product is to recycle the NADH plus. Without recycling the NADH or the NAD plus, this whole process of glycolysis cannot continue. So it's just trying to recycle that NAD plus so that it can accept more electrons so that glycolysis can continue to occur. All right, so this is just reviewing respiration versus fermentation. So again, same first step. Glycolysis is the same, taking glucose, making pyruvic acid, producing ATP and NADH. In cellular respiration, then those pyruvic acids will then go into the Krebs cycle, breaking down into carbon dioxide, producing all of these NADHs, which are going to carry those electrons, yada, yada, right? Whereas in fermentation, we just take the pyruvic acid, convert them into final end products, which will recycle the NAD+. Lastly, we're just going to wrap up by talking about the diversity or the metabolic diversity amongst all these different organisms. So this is the names that you guys need to know over here. And there's basically 3 parts to them. The first part is talking about the source of energy, where that comes from. That can either come from light, which is going to be called photo, or it can come from different chemical compounds, so then it'll be called as chemo. Second part of the word is talking about the source of carbon. So if it comes from carbon dioxide, then it's called auto. If it comes from organic chemicals, then it's going to be hetero. And then the third part is just troph. So for example, we are chemoheterotrophs, which says that we get our energy from chemicals. We also get our carbon from chemicals, which is food that we eat. Okay, so that's where we get all that from. Bacteria, though, can do all of these. Different species of bacteria can carry out different metabolic pathways, can utilize different sources of energy. So they're very diverse. And that's it. That's all we have for chapter 5. I hope you've enjoyed. I know it may be a little bit dense, so please feel free to email me if you have any questions. Stop me if you have any questions. And happy studying.